My name is Mike Denne. I'm a PharmD by training. I lead our U.S. Medical Affairs team at Takeda, focused in rare disease and plasma-derived therapies. I've been working in rare hematologic disorders for over 10 years now, and in the rare disease space for a bit longer than that. TTP, and specifically today we're talking about the congenital form of the disorder, is a genetic disorder that we see happening. It's an ultra-rare disease happening in less than 2 people out of a million, somewhere between 0.5 and 2 people out of a million. And this is a disorder that's resulting from the absence or very low levels of an important enzyme called ADAMTS13. Now, ADAMTS13 plays a really important role in our bodies, and the role that it plays is cleaving an important blood clotting factor protein called von Willebrand factor. So in the absence of ADAMTS13, our von Willebrand factor gets released in a form called ultra-large multimers. And those are important in and of themselves, but you don't want all ultra-large multimers all the time. They're long and platelet rich, and they have a tendency to form these microthrombi, which get stuck in our small blood vessels and lead to ischemia and organ damage in those areas. And so what ADAMTS13 does is it cleaves our von Willebrand factor into monomers and multimers of different sizes, which is functionally helpful for our clotting system. Until the availability of recombinant ADAMTS13 or Adzynma, which we're talking a bit more about today, most of these patients were managed with plasma infusions, either fresh frozen plasma or solvent detergent treated plasma. The plasma infusions can be quite effective. There are some challenges with that therapy. One being that there are a number of people who can't tolerate them and have allergic reactions or infusion reactions to those treatments. Another challenge with current therapies are the duration of administration, where it takes upwards of 2 hours for these infusions to be given once a week or every other week for the patients who are being treated with prophylactic use of the therapies. And it's worth mentioning also that the plasma infusions have a fairly low amount of ADAMTS13, so you're getting a fairly large volume of replacement therapy for a small quantity of ADAMTS13. Recombinant ADAMTS13, which is called Adzynma, is a replacement therapy, specifically of what's missing in this case, the natural ADAMTS13 in congenital TTP patients. And so this therapy can be administered every other week or weekly as needed, and serves to actually replace the missing enzyme at levels approaching that or achieving normal levels immediately after the infusion, which overcomes some of the challenges that we see with the previously used therapies of high volume, lower amounts of ADAMTS13, and the infusion reactions and allergic reactions that we see with some of those patients. So Denne was studied in a phase 3 study that evaluated patients from age 0 to 70. And the study itself was designed with 3 periods. Patients entered the study and got either 6 months of Adzynma or 6 months of their current standard therapy. After that 6-month period, they switched over. The group that had Adzynma received standard therapy. The group that received standard therapy in the first period got Adzynma in the second period. And then all patients entered a third 6-month period where they received Adzynma for that entire time. And so as we talk about the results, we'll be talking about them in the spirit of, was it period 1 or 2 or in period 3 when all patients were receiving Adzynma. The endpoints in the study, the primary endpoint was looking at acute TTP events, which were defined by certain lab parameters, either a reduction in platelets, an increase in LDH above a certain level. And then we also looked at subacute events in the entire study, which were the same parameters, but with less severe changes in those lab parameters, either compared to normal levels or the baseline levels of those patients. The entire study enrolled over 40 patients. The first subset of the patients, which are the first 32 patients, which consisted of adolescents and adult patients, were actually published earlier this year in a New England Journal article and actually served as the patient population for submission to FDA and eventual approval of Adzynma. What we're talking about or have talked about this week at ASH are the continued publication of the pediatric subset of those patients that continued through the study. So we were talking about 8 patients in the phase 3 study between the ages of birth and 12 years, as well as 6 additional patients in a phase 3b study that serves not only as a continuation study, but allows new patients to enroll. So there were 6 unique patients that enrolled into that phase 3b study between the ages of birth and 12 years old. And so the results of this continued pediatric population were very similar to what we saw in the adolescent and adult population in the large group. In the phase 3 study, there were no acute events that occurred in the treatment population, and there were subacute events that occurred at a rate of 0.2 for those treated in the ADAMTS13 arm, and 0.51 for those who were treated with the current standard of care. So results very similar to what we saw in the original study. No new safety events were identified. No patients in the Adzynma arm stopped therapy or had an interruption in therapy as a result of adverse events. And importantly, there were no neutralizing ADAMTS13 antibodies identified in the study. Well, we'll continue to complete this study, and we'll see these patients, or many of them, roll into the 3B study. And so we do expect to have continued data stream looking at safety and efficacy in the population. The original approval of Adzynma included pediatric patients because at that time we knew the pharmacokinetic data, which showed that there were very small or no differences in pharmacokinetics in the pediatric population as compared to the adolescent and adult population. So this new clinical data will be added to that prior pharmacokinetic-based approval and give hopefully our clinicians even more information to use as they're treating these patients with congenital TTP. At Takeda, we continue to be very focused in rare disease, and we have a commitment to these patients. Our goal is to make sure that we're advancing the science and education towards diagnosis and improve patient outcomes in patients with these rare conditions that are oftentimes difficult to diagnose and treat.